Mendel's laws today. Mendel's laws. The laws are written up here. We're going to talk about one of them here and the second one here. We're not going to talk about law of independent assortment. We'll get to that on a later date. This is learning target 5, C, and F. It's learning target C and F. I don't have that written here. But the law of dominance is that one allele is dominant. And if that allele is by itself, then of course that gene's by itself, it's going to show the dominant trait. If it's mixed with a recessive trait, one allele is dominant, one allele is recessive, then it's still going to show the dominant trait. If it is all by itself, both of them recessive, that's homozygous recessive, then it's going to show the recessive trait. So if, in this case, capital R was light blue and little r, or lowercase r, the recessive gene was dark blue, then that's what would happen, just like this. So if you're looking, we'll talk about this will come up in a second, if you're looking at a light blue, whatever this is, it doesn't matter, whatever the trait is, if you're looking at the dominant trait showing through, like this light blue, then you know that there is a dominant trait in each one of them, but you're not sure which one of these it is. It could be either. If you're looking at a dark blue, looking at the recessive trait coming through, then you know that it is a homozygous recessive. It's both lowercase. And the reason you know that is because there's no other way for the dark blue to show. There's no other way for the recessive trait to show unless they're both recessive. And then we get to the law of segregation. The law of segregation has nothing to do with civil rights. It has nothing to do with relations in the mid-1900s. Although that segregation, it does mean the same thing, that maybe where you've heard it from, it is talking about separating alleles. So alleles separate during gamete formation. And this process should be familiar to you. We've drawn this process a lot. You start off, there would actually be a lot of times there'd be another circle up here with a single chromosome, and then S phase happens, it replicates, it splits again. This is PMAT, and then again PMAT, cytokinesis. This is meiosis. This should look familiar to you. It's the formation of gametes. And when gametes, things like sperm cells or egg cells, sex cells, germ cells, all those things, the formation of those things looks like this when we draw it out. In the original cell, it had the upper part was and according to this drawing, it was a heterozygous. It was heterozygous. So there was a gene that was dominant, there was a gene that was recessive, so it was heterozygous. And this law of segregation is just saying that they are separating. You can't have both in one. So one sperm cell is going to have a dominant trait, maybe, and then this over here is going to have a recessive trait. And a dominant and recessive allele for a specific trait. I misspoke. And so they're not going to have both. They can't donate both. They can only donate one or the other. Now, if this up here was all red, and it was all capital R's up here, then that's all they have to donate. But in this case, it was heterozygous, where they could have donated one or the other, and that's what it did. So law of segregation, again, if you can do a Punnett square, if you've already been able to do a Punnett square, you understand a little bit of this. This is why we have a column on the left side, and then a column on the right side, a column on top, and a row on top, row on bottom. It's because this may be the dad's genes. Each of these are a different allele in his DNA, but he can only donate one, and that's what these Punnett squares show. In law of dominance, we see that because we talked about recessive and dominant traits or alleles that manifest in a trait, and I think both of these should hopefully make sense to you already, but you just didn't know that there was a law of dominance and a law of segregation. All right, Mendel's law. Test cross. So over here, we're going to start looking at what a test cross is. A test cross is a way of figuring out what genotype this would be in this situation. If we know that a plant, let's just give an example for something that Mendel would have done. If he figured out that the tall pea plants are going to be a dominant allele, dominant trait, the short would be a recessive trait, a hidden trait, only showing when it's by itself. So if you see short in the trait, then you know that there's going to have short alleles. It's going to have a little t, homozygous recessive genotype. This one over here, like I hinted earlier, this one over here is going to, you're just not sure. It's tall, and you don't know if it's homozygous dominant or heterozygous. You're not sure which one of these it is. So what we're going to do is we're going to use Punnett squares to figure this out. So set up your Punnett square. We'll set up big T with little t right here. And we'll set up big T with little t here. And the data that this gets us should tell us which one this is. So if we put these together, if we breed, if we cross-pollinate this plant and this plant, and it happens to be big T, then the results are going to be this. It's going to, all the children, all the offspring are going to be heterozygous, 100% of them. 100% of them will be tall, and 100% of them will be heterozygous. They'll have 0 short. This is called the genotypic ratio. That's how we write that. And this one, if we were to take big T, little t, which, again, this one might be heterozygous, big T, and we cross it with little t, it would be this Punnett square. And this Punnett square gives us different material, different data. So this Punnett square would give us half tall and half short. It would be none of them are homozygous dominant, 2 of them are heterozygous, and 2 of them are homozygous recessive. Half are tall, half are short. So if you were to take these 2 and breed them together and cross them, and you were to take 100 of the seeds that came off of those plants and then put them in the ground and grow them, and all of them were tall, you would know that this tall one is actually homozygous dominant. If you were to plant it and it was half of them were tall and half of them were short, or something similar like 40-60, if you were to see numbers like that, then you would know that this tall one is actually heterozygous. It's showing the gene for tall, but it actually carries the gene for short, so it can have short babies. And that's what a test cross is. A test cross is, hey, here's an experiment, mental experiment. Let's see if we could figure out what this one is. We know this one, but let's see if we can figure out what this one is. That's a test cross. That's it. And then we got a 2-step cross. A 2-step cross is where we take generation. We cross 2 parents together. We're taking 2 genes. In this case, let's just say that it's still tall and short. You're taking a tall homozygous dominant for tall and homozygous recessive, which is short, and you're crossing them. These are the original parents. We cross them, and then we get the offspring are all heterozygous. That's very similar to what we had up here. They're all heterozygous. This is called the F1 generation, and 100% of these offspring are going to be tall. They're going to show the dominant trait. They're going to have those alleles. And then we take any 2 of these, and in this case they're all the same, but if we were to take any 2 of these and cross them, that would be the F2 generation that we're making. So in that case, it really doesn't matter which one we move because they're all heterozygous, big G, little g. So we move them over, and then we cross them. We cross this would be the dad, this would be the mom, and we cross them. Then we get homozygous dominant, heterozygous, homozygous recessive. And we can do a genotypic ratio of that. And genotypic ratio is always going to be the most dominant, so like capital, to the least dominant, lowercase, and in between that is a heterozygous. Genotypic ratio of this, how many big G's do we have? We have 1. How many big G, little G's, heterozygous? We have 1, 2. And little g, we have little g, we have 1. So the genotypic ratio is 1 to 1, 1 to 2 to 1. And the phenotypic ratio is going to be how many are tall. So this one would be tall because it has the dominant gene. This one is tall because it has a dominant gene, and this one is tall. As far as the phenotype is concerned, all these are exactly the same. And then you have 1 recessive, 1 short one. That's why it's a 3 to 1 ratio. So these are Mendel's laws. Mendel was a very well-known, he's called the father of genetics, very instrumental in helping us understand this stuff, and we're going to go into more depth later on, both into what the law of independent assortment is and other actually non-Mendelian traits that get passed on. So that's for another day.